Hi, today I'll be presenting the basic structure of Cupriatus metallidurans genome, a bacteria known for its survival skills in heavy metal contaminated areas as well as in ultra-clean environments. Its ability to harvest gold from ionic solutions and to withstand normal growth in space. Cupriatus metallidurans has a genome composed of two large circular chromosomes, CHR1 and CHR2, of approximately 3.9 million bp and 2.58 million bp each, and two megaplasmids pmol28 and pmol30 with 171.46 thousand bp and 233.7 thousand bp each. The GC content of the replicons are all a little over 60%. Chromosome 1 carries most of the essential genes, as those needed for DNA replication and repair, cell division, transcription and translation, along with sets of genes required for protein processing, folding, and post-translational modification. There are 4 sets of 5S, 16S, and 23S ribosomic RNA genes, 12 structural RNA genes, and 62 transfer RNA genes, most located on CHR1. Genome analysis predicts 6,717 coding sequences. Of those, 4,518 sequences were assigned a function. CHR2 harbors genes involved in specialized metabolic and biosynthetic activities and adaptive responses, as well as genes for motility and chemotaxis, peroxide removal and oxidative stress, DNA adenine methylation, DNA repair, and adaptive mutagenesis. A striking feature of strain CH34's genome is the presence of at least 16 laterally acquired genomic islands, and the abundance of transposase and recombinase-related gene sequences across the 4 replicons. 11 genomic islands were identified on CHR1 but none on CHR2. Both megaplasmids pmol28 and pmol30 carry key genes for resistance to heavy metals. Plasmid pmol28 encodes resistance to nickel, cobalt, chromate, mercury, and thallium. Plasmid pmol30 specifies resistance to zinc, cadmium, cobalt, copper, silver, and lead. Metal detoxification in CH34 mainly occurs via ion efflux, with 155 genes promoting 30 efflux systems. 3 protein families assure such systems: the resistance, nodulation, cell division family of permeases, the cation diffusion factor family of heavy metal transporters, and the P-type ATPase family of ion pumps. At neutral pH and oxy conditions, Cupriatus metallidurans synthesizes and secretes soluble iron-complexing compounds for utilization of low-solubility iron-3 minerals, called siderophores. On CHR2 are located genes for a unique type of siderophore called staphylophorine B. CH34 has the ability to regulate motility through the formation of biofilms using quorum sensing, with genes located on CHR2 inside a specific operon. It also has over 150 genes involved in generating and controlling flagella, fimbriae, and type 4 pili. CH34 has mechanisms for both aerobic and anaerobic respiration, having the ability to reach full denitrification using nitrate as a terminal electron receptor. It also has multiple genes to assimilate a variety of B vitamins, such as vitamins B1, 2, 5, 6, 7, 9, and 12. In absence of heterotrophic carbon sources, Cupriatus metallidurans CH34 can switch to an autotrophic lifestyle by fixing and reducing carbon dioxide for synthesizing organic molecules, making this through the Calvin-Benson-Bason cycle, for which it has all the necessary enzymes. It can also use the reductive acetyl-CoA pathway, having functional genes to fix carbon dioxide in the Wood-Longdale pathway, with a key biochemical reaction being the transition between carbon dioxide and carbon monoxide mediated by a metalloenzyme. This bacteria also has the capability to oxidate thiosulfate, sulfide, and sulfides. Such mechanisms, combined with an autotrophic lifestyle, would enable Cupriatus to use reduced sulfur compounds as electron donors while using carbon dioxide as an electron acceptor, working as a true chemolithotrophic bacteria. Cupriatus metallidurans can gain energy from oxidizing hydrogen gas into electrons and protons. This ability to use hydrogen instantaneously has a reducing power and hence as an alternative source of energy indicates chemolithotrophic growth as a part of CH34's lifestyle. Cupriavid's metalliduran CH34 can utilize various recalcitrant organics as sole source of carbon and energy, including volatile compounds in the monoaromatic hydrocarbons benzene, toluene, oxyene, and phenol, all highly toxic for humans. Bacteria also produce polyhydrobutyrate, a polyester polymer belonging to the family of polyhydroxyalkanoates, as a carbon and energy reserve to be metabolized after exhausting all the carbon sources, and the main genes for its synthesis in Cupriavidus are divided between CHR1 and CHR2. The robustness of Cupriavidus metalliduran CH34 in harsh environmental conditions does not rest just on its efficient transport and detoxification of harmful substances, ions or molecules, but also greatly depends on safeguarding its genetic material from irreversible damage. This bacteria also has the genes for DNA repair through base excision, the complete pathway for nucleotide excision repair, direct DNA repair, mismatch repair, and responses for heat shock and oxidative stress. Finally, CH34 also harbors 15 proteins belonging to the reversal stress protein superfamily. After all this, Cupriavid's metallidurans is also known to withstand concentrations of silver and gold ions toxic to most bacteria. It's studied as a possible industrial producer of green plastic made of PHB, and it can grow even in ultra-clean spacecraft assembly rooms. Its survival capacity is simply outstanding.